you to my presentation, Biochemistry behind the luminescence of firefly. I'm going to speak a very little only today because I don't know much about the biochemistry behind the luminescence of firefly. So my presentation include introduction, biochemical reactions, mechanism of bioluminescence of firefly, then biosynthesis of luciferin. Luciferin is a key organic compound that plays a major role in the luminescence of these small cute organisms. So far around 2100 species of light emitting bugs or fireflies are reported. Actually these are not comes for some other species are also there. So true fireflies actually comes in the family of Lampyridae, and in this totally in 2100 means around 3 different families are involved, but in general whatever we are seeing the fireflies that is comes in the family of Lampyridae, and these fireflies produces a light or that comes in the red region or in the green region. In general, I have seen so far only green light emitting fireflies. So these organisms are very classic or very much common examples of bioluminescence, where these organisms utilizes chemical energy that is converting into light energy. Let us see how these organisms are able to produce the light inside their body in detail. Here, these organisms produce light by oxidation of an organic substrate called D-Luciferin, and this reaction is catalyzed by an enzyme called luciferase. Here you can see the crystal structure of the enzyme D-Luciferase, which is a photoprotein that catalyzes the light producing reactions. And here you can see the D-Luciferin structure, where you can see an asymmetric center here and with a carboxylic acid attached there. If it is in T form, then only it is able to produce, that compound is able to produce the light, and because that is only having only will bind with enzyme properly, and in its L form it is not able to produce the light. What is happening? It is an enzyme catalyzed reaction, and where a molecular oxygen that is reacting with this organic compound and oxidizing that organic compound named D-Luciferin to oxy-luciferin. This is the general outline of the reaction. Luciferin undergo oxidation by reacting with a molecular oxygen inside the enzyme luciferase, and it will react with in this reaction it will utilize an ATP also, and it will form the oxidized luciferin named as oxy-luciferin, and it will generate a photon. That's a light in the green region around 530 emission. So let's go to the detailed biochemical reaction. What is happening exactly? This luciferin reacts with luciferase. This reaction is happening inside that big enzyme luciferase. So the luciferin bind with the enzyme first, luciferase enzyme first, then an ATP is consumed and an inorganic pyrophosphate is released. Then this AMP, adenosine monophosphate, that will bind with that D-Luciferin and it will form a complex. Later, a molecular oxygen undergoes reaction with this AMP luciferin complex, and this AMP luciferin complex will become unstable upon the presence of oxygen, and an AMP is released from it, and again a new cyclic oxygen, that cyclic compound is produced. This compound is highly reactive, so this is unstable, which will liberate a molecule of carbon dioxide, and a excited state oxy-luciferin complex is produced. This, since this is there in high energy state, excited state, so this want to come to its ground state, it will produce a photon of light for coming back to release the energy. It will produce, because this is in excited state, it is a high energy state. To come back, for coming back to its ground state, this molecule will produce a photon of energy, that is light, and it will come to its ground state. So this oxyluciferin compound in ground state is produced, and this is how fireflies generate light. And how this D-Luciferin is synthesized inside the firefly. That's having a compound named arbutin. This arbutin undergoes hydrolysis and oxidative reactions, and it will generate a benzoquinone structure, parabenzoquinone structure. And this parabenzoquinone structure, upon reacting with two cysteine amino acid residues, and it will produce a either D-luciferin or L-luciferin. Thank you.